Microscopy. 1. Microscopy. Technique to view objects invisible to the naked eye. 2. Discovery and study of cells progressed with microscope invention. Light microscope. 1. Laboratory microscopes are light microscopes. 2. Light microscopes use visible light and a two-lens system. 3. Also known as compound microscope. Components. 2 lenses. Ocular lens. Objective lens, revolving nose. Working principle. 1. Light source illuminates the object. 2. Objective lens forms a real, inverted, and magnified image. 3. Eyepiece uses this image as an object. 4. Eyepiece produces a final, virtual, and magnified image. 5. Final image is inverted with respect to the object. Magnification. 1. Magnification increases object size beyond original size. 2. Represented by x, e.g., 10x, 100x. 3. Calculated by multiplying objective lens value by ocular lens value. 4. Maximum light microscope magnification. 1500x, 100x objective, 15x ocular. 5. Electron microscope magnification. 1 to 50 million times.
Diagram of my. Dear, just like and subscribe my. Resolution. 1. Measures ability to distinguish between two close points. 2. Naked eye resolution, 0.1 mm. 3. Light microscope resolution, 250 nanometers. 4. Electron microscope resolution, 0.2 nanometers. 5. Resolution isn't improved by increasing magnification. Difference between magnification and resolution. Magnification makes small objects appear large. Resolution distinguishes between two objects. Electron microscope. 1. Uses beam of accelerated electrons to visualize specimens. 2. High resolution, 0.2 nanometers. Working principle of electron microscope. 1. Electron gun produces beam of 2. Condenser lenses focus electrons on specimen. 3. Accelerating potential, filament anode, moves electrons down. 4. Specimen, thin section, 20 to 100 nanometers, on specimen holder. 5. Denser regions scatter more electrons, lighter regions scatter fewer. 6. Scattered electrons pass through objective lens, magnification. 7. Ocular lens further magnifies image. Comparison between light microscope, electron microscope. 1. Light source, visible light, 400 to 700 nanometers. 1. Light source, electron beam, 1 nanometer. 2. Magnification, 1500x. 2. Magnification, 1 to 50 million x. 3. Specimens, live or dead. 3. Specimens, dead or dried. 4. Image formation, light absorption. 4. Image formation, electron scatter. 5. Resolution, low, 250 nanometer. 5. Resolution, high, 0.2 nanometers. 6. Useful magnification, 500x. 6. Direct magnification, 16,000x. Photographic, 1 million x. 7. Image, colored. 7. Image, black. 8. Vacuum, not required. 8. Vacuum, essential. Thank you. Like and subscribe my channel.